Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor from Department of Biochemical Engineering and Biotechnology. Today we are going to talk on the module called B cell surface molecules from the paper Immunology. Now, lymphocytes are of two kinds, B cells and T cells. Both evolve from the stem cells. For both cells, there are two broad phases of cell development. One phase is antigen dependent, another phase is antigen independent. Today we will focus more on details of the antigen independent phase of the B cell development, because that normally precedes the second antigen development phase. So the objectives of this module can be said to be to understand the early phase of the development of the B cells and the role of bone marrow stromal cells, which play a very crucial role at this stage. We will also understand the role which is played by the gene segment recombination at these various stages. We will also look at the relative roles of the bone marrow and peripheral tissue in these developments. So the concept which are involved are that how do the stem cells, hematopoietic stem cells, they become first early pro B cells, then they become late pro B cells, then these late pro B cells develop further and become small pre B cells. These then in turn become immature B cell, and finally we have the mature B cells. So while discussing clonal selection theory, we had discussed some features like how clones are selected from the pre-existing ones. In this discussion, today we will look at the creation of these clones of B cells from the stem cell which is present in the bone marrow. The essential initial step in the adaptive immune response is the interaction of B lymphocyte cell surface with an antigen. The exact nature of this contact, though may be different when macrophages and T cells are present. Nevertheless, the contact changes the nature of the gene expression in B lymphocytes. A chain of events starts which results in multiplication and differentiation of B cell lymphocytes. Before this, B lymphocytes were already specialized cells in the sense that these had the capacity or the potential to respond to the presence of antigen. So their differentiation finally results in their becoming plasma cells secreting antibody specific to antigens or becoming memory cells. Let us look at the B cell differentiation in fetal liver and then in bone marrow. What is shown in these figures, on the left hand side there is a section of the bursa showing B cells developing in follicles. We have already mentioned bursa in the birds is a distinct organ which is involved in the B cell differentiation. In humans, there is no bursa. On the right hand side is the section of the human fetal liver showing islands of hemopoiesis. By about 8 to 9 weeks of pregnancy in women, B cell precursors can be identified in fetal liver. In mouse, this period is of about 14 days. Later, production of these B cell precursors gradually stops and this process is taken over by bone marrow. Our insight into the stages of B cell differentiation has come from studies on various animals in the embryonic stages. There are some similarities in these steps in mice, chickens, and humans. As we pointed out, in the chicken, the site of B cell lymphopoiesis is a lymphoepithelial organ called the bursa of Fabricius. In the early days, many studies were devoted to identifying bursa or its equivalent in mammals. Ultimately, it was established that mammals do not have bursa or an equivalent organ. In mammals, it was found that B cells develop from the lymphoid stem cell in the fetal liver. In chicken or quails, stem cells appear into bursa by about 10 to 14 days of life of embryo. In other systems, bone marrow takes over differentiation of B cells. This is true of other cells as well, for example, erythrocytes, granulocytes, monocytes, and the blood platelets. Their differentiation also takes place in bone marrow. Let us look at what happens in the bone marrow vis-a-vis B cell differentiation. While on the left hand side you have an actual picture of bone marrow where all this takes place, on the right hand side a model is sketched out showing that what are the various stages of B cell differentiation in bone marrow. Stromal cells of bone marrow play crucial role in the development of B cells. The word stroma is derived from a Greek word which means mattress. B cells taken out of bone marrow fails to grow in vitro. Hence, support of stromal cells is necessary. In other words, stromal cells provide some kind of a physical support as well as the necessary interactions for this differentiation to take place. These stromal cells are mixture of many phenotypes and resemble fibroblasts, endothelial cells, and smooth muscle cells. These stromal cells produce collagen, laminin, and actin. Stromal cells are believed to support B cell development by not only providing specific contacts but also via growth factors which are responsible for multiplication as well as differentiation. Stem cell factor SCF is one such growth factor which is a membrane bound cytokine on stromal cells. It interacts with tyrosine kinase present on the surface of precursor of B cells. The stromal cell derived factor or the pre-B cell growth stimulating factor, which is called either SDF1 or PBSF, is also required. Mice which lack the gene for this factor fail to produce developed B cells. The role of SDF1 is believed to be in retention of precursor B cells in the marrow. At some stage, interleukin IL-7 is also required for B cell development. During development, B cells migrate within bone marrow, becoming more independent of the stromal cells. More than 75% of B cells which mature in bone marrow undergo apoptosis and undergo phagocytosis by macrophages present within the bone marrow. For example, murine B cells are produced at the rate of 5 into 10 to the power 7 per day. However, murine spleen at any time contains around 7 into 10 to the power 7 B cells only. Apoptosis or development into mature plasma cell are not the only two fate of the precursor B cells. B cells which produce receptors on their surface which react with self antigens either die or are inactivated or undergo receptor rearrangement. If the self antigen is multivalent, apoptosis is the dominating outcome. This is called clonal deletion. In other cases, receptor editing leads to a new receptor. Some other cells may become unresponsive to the antigen. These cells are called ignorant cells. Such ignorant cells are not able to sense self antigen either because it is in low concentration or inaccessible due to sequestration or is unable to cross-link the B cell receptors. The various fates of the immature B cell are shown in this diagram. The B cell development is characterized by appearance of cell surface proteins. What we have shown here is the development of a B lineage cells proceeding through several stages marked by the rearrangement and expression of the immunoglobulin genes, that is H chain genes, L chain gene and surface immunoglobulin gene. Pro B cells are progenitor cells with very little capacity for self renewal. These are first identified stage next to pluripotent stem cells. Early pro B cells are identified by joining of DH to JH. Late pro B cells contain VH joining DJH. The large pre-B cells have complete mu chain expressed intracellularly. Some mu chain may start appearing on the cell surface. In such cases, the mu chain appears in combination with a surrogate light chain. This together is called pre-B cell receptor. At small pre-B cell stage, heavy chain segment rearrangement is stopped. Expression of surrogate light chain also stops. The mu chain occurs only intracellularly. The next stage in the B cell development is the immature B cell. This contains IgM on the surface. All these stages of B cell development are antigen independent stages. In mammalian fetus, all these stages occur in liver and spleen. After birth, all these stages of development takes place in bone marrow. Pre-B cells are seen in human fetal liver by 7th or 8th week of gestation. By 9th week of gestation, B cells in fetal liver have surface receptors for the complement and IgM. By 10th and 12th lymphocyte expressing other Ig class are present. By 15th week, distribution of B cells in the blood, spleen, lymph nodes and those expressing different Ig isotypes is similar to an adult. While IgM synthesis can be seen by 10th or 12th week of gestation, Ig synthesis takes place later. Serum and secretory IgA is detectable only by 13th week. The neonatal serum contains no IgA, only small amounts of IgA, M and maternal IgG in neonates. The IgM response is longer lasting than adults. IgG and IgA responses are weak. This is presumably due to lack of T cell cooperation. Neonates also lack IgG2 subisotype and thus are unable to respond to carbohydrate antigens. Cells now move over to secondary lymphoid tissues. The ones which survive there further develop into B cells. These various stages of development of B cells in the bone marrow are nurtured by stromal cells and various cytokine and growth factors as already discussed. These naive B cells recirculate through peripheral lymphoid tissues only when clonal is selected after they encounter the specific antigen. They become plasma cells which actively secrete IgM initially and then IgG. Apart from appearance of these immunoglobulin or their chains, these stages of development are also accompanied by display of various other cells surface proteins. Cell surface markers are designated by acronym CD, which stands for cluster determinant, followed by a number. These numbers have no physiological significance but merely indicate their order of discovery. Anti-CD4 implies an antibody for CD4 cell surface antigen. One of the earliest such marker is CD45R. This is a variant of CD45 which has been also called common leukocyte antigen. This is because variants of CD45 also appears on the surface of T cells, monocytes, neutrophils. CD45R is seen in pro-B and in all subsequent B cell stages right up to plasma cell. It is a tyrosine phosphatase in mouse. The nomenclature there is different and equivalent of CD45R is called BT220. CD19 is another protein which is present at all stages of B cell development. Both CD45R and CD19 participate in the B cell receptor signaling. B cell receptor signaling is important for the development of B cells. So, all B cell precursors start assembly of CD45R and CD19. Similarly, receptor for IL-7 is expressed on the B cell precursors. This interleukin is essential factor for the growth of both B cells and T cells. Use of anti-IL-7 antibody or mutants in which IL-7 is inactivated stop B cell development, which unequivocally establish the role of these cell surface markers. CD43, also known as mucin leukocyalin, appear at pro-B cell stage but is not there on immature B cells. CD43 is an adhesion molecule participating in interaction of B cell precursors with other cells. One such important interaction is with the stromal cells. KIT, the receptor for SCF1, stimulates both lymphoid and myeloid developments in the bone marrow. It is turned off at the late pro-B stage. At large pre-B cell stage, CD25, a low affinity IL-7 receptor, is expressed. KIT and IL-7 are involved in pro-B and pre-B cell proliferation. What is the role of rearrangement of gene segments for immunoglobulins? The assembly of V gene segment by recombination is an imprecise process as random addition of nucleotides around the joining points is involved. We may recollect that this is a built-in strategy for creating diversity of antibodies specificities. It is estimated that about 2 out of 3 events generate out of frame sequences. These non-productive joins have no use. B cells which end up making such non-productive joins are eliminated. Let us look at the phenomenon of allelic exclusion to which we had briefly referred to. A B cell either uses the genes from the paternal chromosome or from maternal chromosome. Once an IgDNA segments are successfully rearranged, the cell is committed to express that particular V regions on both L and H chains. Other V gene segment rearrangements are not allowed. If the right gene arrangement does not take place, the B cell tries it again till it gets it right. If the cell runs out of option, it stops. Similarly, once it gets it right, it stops and no further rearrangement takes place. If VH rearrangement is right, no further VH rearrangement takes place. Similarly, if the right VL rearrangement takes place, no further VL rearrangement takes place. This cell and all its progeny are committed to produce Ig with the V region and hence with same corresponding specificity. This figure again shows 4 different productive rearrangements of a V kappa and J kappa gene segments. Only one strand of the DNA is shown for simplicity. The 4 different in-phase joining of the 2 gene segments produce 3 different amino acid sequences at the seam between the 2 segments. This we had discussed earlier. It is important to recall this again in this context to indicate that successful or productive rearrangement implies expression of a functional polypeptide. Non-productive rearrangement implies an incorrect reading frame or creation of a termination codon at downstream to the joining parts. Non-productive rearrangement seldom leads to the formation of messenger RNA, but polypeptide expression is very rare. This figure shows the examples of junctional diversity arising from the joining of the JH and D gene segments to form VH genes. In the A part of the figure, the joining of the D gene segments to the JH1 gene segment is shown. In the B part of the figure, the joining of homologous D gene segments to different VH segments and the same JH gene segment is shown. During B cell development, H chain rearrangement produce pre-B cells. After that, L chain rearrangement leads to the B mu cells. First kappa chain is rearranged, and only if kappa chain rearrangement is unsuccessful, cell attempts lambda chain rearrangement. This is inferred from the observation that B cells generally do not have lambda chain rearrangement. On the other hand, cells which ultimately express lambda chain do have rearrangement of kappa chains. The signal for the termination of H chain rearrangement takes place before L chain rearrangement is initiated. The termination of H chain rearrangement is seen by insertion of H chain in the endoplasmic reticulum. Appearance of IgM on the cell surface indicate that L chain rearrangement is terminated. This well orchestrated regulation ensures that any B cell produces immunoglobulin with lambda as the light chain or kappa as the light chain. The result is immature B cell, but which is committed to produce antibody only of one specificity. H chain gene rearrangement is initiated at early pro-B cell stage with D gene segment joining JH gene segment. This occurs at both alleles of the H chain. This results in late pro-B cells. Most DJH joining in humans are productive because of the sequence of human D gene segments. Presumably, having two successful DJH rearrangement is set against large failure rate for V2 DJH joining. It is V2 DJH rearrangement takes place only on one chromosome to attempt mu H chain formation. Successful formation of mu H chain leads to pre-B cell stage. In case of an unsuccessful mu chain gene formation, cell tries rearrangement on another chromosome. This is the way allelic exclusion is ensured. The large pre-B cell with the mu chain is now ready to divide and proliferate. Pro-B cells with non-productive rearrangement for producing mu chain gene are eliminated. The transient pre-B cells receptor with mu chain as its parts provides the signal to proliferate. This also ensures that inability to receive the signal leads to elimination of the cells. The pre-B cell receptor is made up of mu chain along with a surrogate light chain. This surrogate L chain consists of two fragments joined non-covalently. One is lambda 5, which is quite similar to C domain of the lambda chain. Another is V pre-B, similar to V domain of the light chain, but has an extra amino terminal region. The mu chain surrogate light chain are also associated with two accessory chains Ig alpha and Ig beta to form the pre-B cell receptor complex. This pre-B cell receptor complex resembles B cell receptor but is expressed at low level and is formed only transiently. The productive rearrangement of a heavy chain and formation of a pre-B receptor complex stops division of the large pre-B cells and they become small pre-B cells. Small pre-B cells initiates a rearrangement of light chain in immature B cell, which leads to IgM display on the surface along with Ig alpha and Ig beta. This sends a stop signal to L chain rearrangement. Thus, we finally have a mature B cell with a defined specificity. These B cells with the functional B cell receptor undergo a check for distinguishing between self and non-self. If the B cell binds with a significant binding constant to a self antigen, it will not mature any further. This is the negative selection process. The positive selection for further maturation presumably come from Ig alpha in mice. B cells which lack cytoplasmic domain in their Ig alpha show 4-fold reduction in marrow and 100-fold reduction in number of peripheral B cells. The Ig alpha possibly plays a role in B cell migrating to periphery and their survival there. To summarize this lecture, we learnt that the stromal cells of bone marrow and some cytokines or chemokines play critical role in early days of these developments, early phases of the developments of B cells. So these stages are stem cell, early pro-B, late pro-B, large pre-B, small pre-B, immature and mature. The stages of developments are characterized by appearance of different cell surface markers. These include receptor complex including immunoglobulin chains in the later stages, B cells which have specificity for self-antigens are eliminated during circulation to peripheral lymphoid tissues. We have already discussed earlier how mature B cells, once clonally selected, become plasma cells when we talked about clonal selection theory. So this discussion has been retrospective in nature to look at the earlier stages of development of the B cells. We also covered how other features of clonal selection theory operate. These include one B cell producing antibody of the same specificity and B cells not secreting antibody against self-antigens. Thank you.